Hi viewers, today we are going to discuss the counter current mechanism in the nephrons which makes the hypotonic urine into the hypertonic hyperconcentrated urine. If you observe the vertebrates, the mammals excrete hypertonic urine. Mammals excrete hypertonic urine. For example, if we observe the human beings, the cytoplasmic osmolarity of the blood is of about 300 units. Osmolarity of the blood is 300 units. Whereas the human beings excrete of about 900 to 1200 milliosmolar concentrated urine. So the blood concentration is of about 300, but the concentration of the urine is almost all hyper, that is 900 to 1200 milliosmoles per the decimeter of blood. So here, whenever if you observe further more organisms in the apart from the mammals, not only the human beings, if you observe the vampire bat which lives in South America and which feeds upon the blood of other mammals and the aves. So it excretes the urine, the concentration is 4600 units. That much hypertonic urine it excretes. And if you see another more example, that is Australian hopping mouse. Australian hopping mouse which lives in the deserts of the Australia, and the concentration of the urine is 9300 units. 9300 units. So this is almost all 25 times greater than the normal concentration of the body fluids. It is almost all 25 times greater than the normal concentration of the body fluids. So that is the medullary tissue of the kidneys of the mammals. Otherwise, the interstitial fluid of the kidney of the mammals, they have higher osmotic gradient and thereby they can capable to excrete, otherwise form hypertonic urine. So if you observe, just now we discussed, so this concentration, interstitial osmotic gradient is 1200 units in human beings, 4600 units in vampire bat, and 9300 units in hopping mouse. Right? In order to form the hypertonic urine, they will maintain the higher osmotic gradient in the interstitial fluids of the medulla. So this is known as the counter current multiplication. Counter current multiplication. Counter means opposite. Counter means opposite. Current means flow. Current means flow. Opposite flow. Opposite flow. Okay. The solutions which are present in the renal tubule, otherwise nephron of the kidney, and the blood vessels of the kidney, they are arranged in a parallel manner, but the solutions which are present, which are flowing in the renal tubule and the blood vessels, they are flow in a opposite direction. That is, arrangement of the renal tubule and the blood vessels in a parallel manner, but the solutes which are flowing in the blood vessels and the renal tubule always in the opposite manner. This is nothing but the counter current mechanism. So in the kidney of the mammals, there are two different counter current mechanisms. They are active and working simultaneously, and they form the hypertonic urine. Let us see what are those two. The first countercurrent mechanism is seen in the limbs of the Henle's loop and the collecting duct, and second one is the vasarecta. Second one is in the vasarecta. Let us see how the countercurrent mechanism helps in the formation of the hypertonic urine in the limbs of the Henle's loop and the collecting duct. So this is the first kind of countercurrent in the kidney. Countercurrent in limbs of Henle's loop and the collecting duct is the first mode of countercurrent. So here the arrangement of limbs of Henle's loop and collecting duct are parallel, but the flow of solutes which are present in these vessels is opposite. Okay, and due to the opposite flow and also due to the reabsorption of the glomerular filtrate, there will be a formation of higher osmotic gradient within the medullary tissue. Okay, countercurrent mechanism is nothing but the opposite flow of the solutes in the renal tubule and the blood vessels. Okay, what it makes? It forms the hypertonic urine by making the higher osmotic gradient in the medullary region. Why? Because the concentration of the osmotic gradient of the tissues is how much it is if you observe the 1200 units in the human beings. Okay, what is the normal blood pressure? Normal osmotic pressure, that is the normal milliosmolarities of the blood is the 300 to 310, but it reaches of about 1200 milliosmoles. 1200 milliosmoles. Okay, how it is being achieved by the medullary tissue is the renal infiltrate, while it is traveling from proximal culminatory tubule to that of the Henle's loop. Okay, the sodium chloride, urea, they are absorbed into the medullary tissue, and thereby there will be enhanced concentration in the interstitial fluids, interstitial area. Okay, we have already discussed in the selective reabsorption the sodium chloride from the Henle's loop and the urea from the collecting tubules. They are reabsorbed and stored in the medullary tissue. We have already discussed in the selective reabsorption. Okay, in order to understand clearly the countercurrent mechanism in the first phase, okay, we have to see how the infiltrate that moves from one region to the another region of the nephron. If you observe, so this brown color one is the nephron, Henle's loop. Okay, so this is the proximal culminatory tubule, Henle's loop, distal culminatory tubule, and this is the collecting tubule or collecting duct. Okay, now if you observe the descending limb of the Henle's loop, descending limb of the Henle's loop is permeable to water, but impermeable to salts. Okay, as and when the infiltrate, whenever it is in the proximal culminatory tubule, whenever it enters from the Bowman's capsule into proximal culminatory tubule, it is only 300. Okay, but whenever it comes to the hairpin loop, it becomes 1200. That is due to the releasing of water from the descending limb. It loses water from the ultrafiltrate and becomes concentrated. Thereby, the milliosmolarity of the infiltrate is priorly 300 milliosmoles, 300 units, but whenever it reaches the hairpin loop, it becomes 1200. It loses the water, but it retains the salts like the NaCl. Why? Because the descending limb is permeable to water but not the salts. Okay, thereby it is highly concentrated and concentration reaches 1200 osmolarity. Okay, now the NaCl is ready to relieve from the loop. Okay, then if you observe the ascending limb of the Henle's loop, ascending limb of the Henle's loop, this ascending limb is impermeable to water, but permeable to ions, otherwise salts. Impermeable to water but permeable to salts, so that the amount of NaCl which is present in the infiltrate, that comes out and pushed into the medullary tissue. It pushed into the medullary tissue from the ascending limb. This is always takes place, that is removal of the NaCl. It is always takes place by energy dependent sodium potassium pump. It always takes place through the energy dependent sodium potassium pump. And due to the accumulation of salts within the medullary tissue, there will be enhancement of the concentration. And now the main important thing occurs, the descending limb of the Henle's loop, it pulls the water from the collecting duct. It pulls the water from the collecting duct towards the descending limb, and thereby the water which is released from the collecting duct, that mix up with the blood vessel, otherwise the vasarecta. Right? Whenever there will be accumulation of the salts in the medullary tissue, there will be increase of the osmolarity, and thereby the descending limb of the Henle's loop, okay, it attracts or pulls the water towards the descending limb from the collecting tubule, and the water which is released from the collecting tubule, that mix up with the capillaries of the vasarecta. Okay, although that is whenever the water released into the medullary region, there should be the decrease in the concentration gradient, but here that will not happen. Why? Because whatever the amount of the water which is released from the collecting duct, that mixes with the vasarecta only, mixes with the blood of the vasarecta, so that there will be a maintenance of the osmotic gradient. There will be maintenance of the osmotic gradient, although there will be release of the water from the collecting tubule. Okay, then thereby the little hypotonic urine which is formed in the Bowman's capsule, that is becomes hypertonic. So whenever it reaches the for example, if we start from here, whenever it reaches the hairpin loop, it is 1200 hypertonic, and whenever it reaches the distal convoluted tubule, it is becoming 100, but at the end of the collecting tubule, again it attains 1200. So removal of water, it becomes concentrated, it get concentrated. Okay, and release of ions, salts, it becomes hypotonic, and again by releasing of water, it become hypertonic. Okay, now the countercurrent mechanism which is formed in the Henle's loop, okay, there will be a major role played by the collecting duct. Why? Because the collecting duct also parallelly arranged, to the ascending limb and the collecting duct, both of them parallelly arranged, but the solution flow is somewhat opposite. Okay, so At the end, what we have to remember here, the hypotonic urine, which is in the form of primary urine, ultrafiltrate with the less osmolarity. By losing water, it becomes hyper concentrated at the hairpin loop, 1200 units. And again, here the water is impermeable, thereby the salts removed out, so that the concentration decreased up to 100 units in the distal convoluted tubule. But if you observe furthermore, due to the activity of the ADH on the collecting tubule, what happens? There is a facultative water reabsorption, so that the hypotonic urine, which is containing 100 units and 300 units of osmolarity, that becomes, due to the removal of water, it becomes 600, 900, and finally it is achieving 1200 osmolarity concentration. That is only due to the removal of water, reabsorption of water from the collecting duct due to the effect of antidiuretic hormone. Okay. Then the Henle's loop that exhibits many characters which are to be present in the countercurrent mechanism. It is almost all similar to the countercurrent heat exchangers of the endotherms, which are useful for the regulation of the body temperature. And the Henle's loop, it performs the functions like a energy consuming mission. Henle's loop, it acts like a energy consuming mission. So this energy is used to maintain the high osmolarity in the kidney tissue, and remaining energy will be used to transport the sodium chloride ions in the ascending limb. Some energy will be used to maintain the higher concentration osmolarity in the kidney tissue, and remaining energy will be used for the transportation of sodium chloride in the ascending limb of the Henle's loop, right? And those systems which are maintaining the concentration gradients, they consume the energy and they maintain the concentration gradients. They are known as the counter current multiplier systems. What are the counter current multiplier systems? The systems which consume the energy and they maintain the concentration gradients in different regions of the kidney. Okay, so if you observe the concentration of cortex, it is only 300 milliosmoles, whereas if you observe the concentration of the medullary region, okay, at the Henle's loop or at the collecting duct, it is almost 1200. So that they are known as the counter current multiplier systems. Okay, so the juxtamedullary nephrons also, they are located in the inner side, deeper regions of the extend up to the deeper regions of the medulla, so that they can form the hypertonic urine. This is all about the role of the Henle's loop and the collecting duct in the formation of hypertonic urine. And the second system of the countercurrent mechanism is present in the vasarecta. So what is the vasarecta? If you see, there is a afferent renal arteriole that enters into the Bowman's capsule, formed into the for example, this is the Bowman's capsule. Okay, and this is the afferent arteriole. This enters into the Bowman's capsule and form the glomerulus and comes out as efferent arteriole. This efferent arteriole, this spreads over the proximal convoluted tubule and distal convoluted tubule in the form of peritubular capillaries network. Okay, now apart from the peritubular capillaries network, there are some other channels. Okay, they fuse to form a loop, and these loops of the blood vessels, they also travel along the ascending descending limbs of the Henle's loop, and they also form the countercurrent mechanism. Here this is the Henle's loop of the nephron, but this is the vasarecta, which is formed by the efferent arterioles. Efferent arterioles. Okay, that is the arrangement of Henle's loop and arrangement of vasarecta blood vessel. They are parallel, one by another, parallel to each other, but the blood flow is always opposite. Okay, the ultrafiltrate flows in this direction in the descending ascending loop of the Henle's loop, but here the blood flows in this direction. Arrangement is but the blood flow is somewhat opposite. Okay, now if you observe the flow of the ultrafiltrate in the descending limb, it always flows from cortex to medulla. Okay, then if you observe the flow of blood in the descending limb, it is from medulla to cortex, but if you observe the blood vessel, the blood flows from cortical region to medulla. Okay, thereby what happens? It receives more and more salts. Okay, and thereby it get concentrated up to 1200. Previously we discussed that the ultrafiltrate also, it releases water, okay, get concentrated to 1200. In order to reach that, here also the blood vessel, it receives more and more NaCl, which is released from the ascending limb and get concentrated to 1200 milliosmoles. Okay, as and when it goes up to the cortical region, okay, water is added upon and becomes 300. The milliosmolarity of the blood is 300. That is the flow of the urine, primary urine in the nephron, that is Henle's loop. The blood flow in the vasa recta, both of them opposite to each other. Here in the Henle's loop, descending limb, the primary urine with the 300-400 milliosmoles, that concentrated to 1200 by removing water, and again it is diluted by removing NaCl and gets 100 milliosmoles. But here the blood, it receives the sodium chloride and the potassium ions into the blood vessel. So here the milliosmolarity 300 raised to 400, 600, 900, finally it achieves 1200 milliosmole concentration. And again it moves towards the cortical region, it adds water which is removed from the descending limb and get hypotonic. So that is the arrangement of the Henle's loop and the vasa recta are parallel to each other. Okay, but the flow of the renal filtrate and the flow of the blood in between these two vessels is quietly opposite. Okay, here as and when the blood flow and the ultrafiltrate flow, if they are opposite to each other, there will be a easy exchange of materials takes place. That is nothing but the counter current mechanism. And another specialty of this is juxtamedullary nephrons are having the capacity to increase the concentration of the urine. This is the adaptation of the terrestrial mammals. That is the mammals, they retain how much amount of water they require, and the excess salt and nitrogen bases will be released out, excreted out through the urine. Okay, that is the The cortical nephrons arrangement and the juxtamedullary of nephrons arrangement is very critical and very crucial for the maintenance of the production of the hypertonic urine. So this is the second kind of counter current which will be seen in the vasarecta and the Henle's loop. And coming to the regulation of excretion, here regulation of excretion takes place in three ways, namely through the ADH, antidiuretic hormone, GGA, juxtaglomerular apparatus, and AN of atrial natriuretic factor or peptide. Atrial natriuretic factor or atrial natriuretic peptide. See one by one. First one is the antidiuretic hormone. Antidiuretic hormone is commonly known as the vasopressin. Commonly known as the vasopressin. Okay. Whenever there is a high, the vasopressin, actually, if you observe the brain, here there is a hypothalamus. The special neuronal cells, they synthesize the vasopressin and release the vasopressin into the posterior lobe of the pituitary gland. Okay, that is the neurohypophysis, and this vasopressin stored temporarily in the neurohypophysis as and when there is a more osmotic pressure of the blood. If there is a more osmotic pressure of the blood, otherwise, if the volume of the blood is less, thereby the ADH get released from the neurohypophysis, right? That is, whenever there is a high osmotic pressure of the blood and where there is a less osmolarity of the ultrafiltrate, that makes the release of ADH from the neurohypophysis. What is the main function of the neurohypophysis is vasopressin is the vasopressin released from the neurohypophysis and act upon the distal convoluted tubule and the collecting tubule, and thereby it enhances the permeability of distal convoluted tubule and the collecting duct, and thereby the distal convoluted tubule reabsorbs more water and collecting duct reabsorbs more water, and thereby there will be a enhancement of the concentration of the urine, and there is a saving of water. That is, diuresis is somewhat a release of more water. So it restricts the water release, water wastage from the urine. So that it is antidiuretic. Antidiuretic. Right? Now, whenever if there is a ADH, the functioning of the kidneys is somewhat different whenever there is no ADH at all. Whenever the ADH is present, antidiuretic hormone is present, during that time the water which is present in the ultrafiltrate, that will be reabsorbed into the cortex and medullary tissues of the kidney, thereby less amount of urine produced. Less amount of urine is produced. Decrease in urine production. Okay. Whenever there is a less ADH is less. During that time, due to the non-reabsorption of appropriate amount of water, more and more water will be excreted out through the urine. Thereby, the urine volume will be enhanced. So, losing of more water through the urine is termed as the diuresis. In the terminology of the medicine, it is called as the diuresis. Okay, ADH stops the diuresis, so that it is antidiuretic. It is antidiuretic hormone. Actually, it is a vasopressin. It is restricts the release of water along with the urine, so that it is antidiuretic hormone. Okay, whenever there is a scarcity or lack of ADH, that leads to the polyuria. Polyuria. It is also known as the diabetes insipidus. Diabetes insipidus. That is polyuric condition. Okay, usually the healthy individuals excrete 1.8 liters of urine. Instead of that, more than 3 or more than 5 liters of urine will be excreted out due to the deficiency of the ADH. Deficiency of the ADH. Okay, then it is somewhat different from the diabetes mellitus. Diabetes insipidus is due to the deficiency of the ADH, and thereby there is a diluted urine will be excreted out. Diluted urine will be excreted out. In order to compensate that, then we have to drink more and more amount of water. And there is another term which is caused due to the deficiency of the insulin, diabetes mellitus, that is dealt with the carbohydrate metabolism, that is glucose metabolism. But here there is no relation between glucose metabolism and the diabetes insipidus. Okay, those people who are drinking more alcohol, there will be imbalance in the water concentration, water levels, and thereby due to the dehydration, they release more and more amount of water along with the urine. They undergo dehydration. Okay, why? Because the alcohol that restricts, otherwise inhibits the activity of the ADH. And other diuretic drugs, they also tend to release more amount of water, and not only that, they also decrease the BP. They also decrease the BP. So this is all about the ADH hormone. Simply, ADH releases from the vasopressin, okay, and that act upon the distal convoluted tubule and the collecting duct and enhances the permeability, and thereby it reabsorbs more and more water into the cortical and medullary regions, and thereby reduce the volume of urine production. Okay, and followed by enhancement of blood pressure. And the second level of regulation on the kidney functioning is done by RAAS, Renin Angiotensin Aldosterone pathway, RAS pathway. Okay, so this is done by JGA, juxtaglomerular apparatus. Juxtaglomerular apparatus. Juxtaglomerular apparatus is present at in between the distal convoluted tubule and the efferent arteriole, right? So this is, so while we are studying the structure of the nephron, we have studied the macula densa and the juxtaglomerular cells. So both of them, they fuse to form the juxtaglomerular apparatus. This juxtaglomerular apparatus, whenever the volume of the blood in the afferent arteriole, otherwise there will be a decrease in the BP, blood pressure, that time this juxtaglomerular apparatus that produce renin. Whenever there is a dehydration and sodium deficiency or hemorrhage, during that time, if there is any decrease in the BP or blood volume, okay, decrease in the BP or blood volume or decrease in the pressure, that leads to the production of the renin by the juxtaglomerular apparatus, right? There are the two conditions that stimulate the production of the renin. One is the decrease in the blood volume, another one is the decrease in the blood pressure. Both of them, they tend to stimulate the release of renin from the juxtaglomerular cells of the kidney. Now this renin, what it will makes, the liver produces angiotensinogen substrate, angiotensinogen. This angiotensinogen undergo catalytic activity by the renin, get modified into angiotensin 1. This angiotensin 1 modified into angiotensin 2 through angiotensin converting enzyme which is produced by the lungs. See, renin is produced by the kidney. We have to remember the source of the hormone. Renin is produced by the kidney. It converts the angiotensin. This angiotensin produced by the liver. Okay, this angiotensinogen finally converted to angiotensin 2. So for the conversion of angiotensin 1 to angiotensin 2, it requires a angiotensin converting enzyme which is produced by the lungs. Okay, thereby this angiotensin 2 acts upon the adrenal gland which is located on the dorsal side of the kidney. Okay, adrenal gland, especially the adrenal cortex, and that leads to the release of aldosterone hormone. As we know, the cortical hormones are cortisone, cortisol, corticosterone and aldosterone. These are the hormones which are produced by the adrenal cortex, whereas the medulla, it produces the catecholamines like the adrenaline and noradrenaline. Now, this aldosterone which is produced by the adrenal cortex due to the stimulation of angiotensin 2, it acts upon the kidneys. Okay, and there will be a increased reabsorption of sodium and water takes place. Increased reabsorption of sodium and water, and thereby the reabsorbed water mixed up with the blood, and thereby there is an increase in the blood volume, and thereby there will be an increase in the blood pressure. Okay, and not only that, there will be a vasoconstriction by the angiotensin 2. Vasoconstriction by the angiotensin 2. Okay, so the decreased blood volume increased and decreased blood pressure increased by the RAS pathway. Okay, that is the renin angiotensin and aldosterone system. It acts like a feedback mechanism, and thereby it balances the urine production. Okay, then come to the third one, the atrial natriuretic peptide or atrial natriuretic factor. Okay, this atrial natriuretic factor produced by the heart, and it acts upon the kidney at the third stage. Okay, whenever there is a more BP in the blood vessels, more amount of blood in the body, in the tissues, then what happens? The natriuretic peptide that produced from the heart. Natriuretic peptide produced from the heart by the stimulation of more BP. Okay, thereby what is the main function of the natriuretic peptide or factor is, it is always agonist to the aldosterone and agonist to the ADH. Okay, this hormone, it inhibits the JGA activity, juxtaglomerular activity, and thereby stops the production of the renin. It stops the production of the renin, and thereby stops the reabsorption of the sodium and stops the release of the aldosterone. Okay, then what happens here? Due to the inhibition of the aldosterone, there will be a inhibition of the sodium reabsorption, and here due to the ADH inhibition, and thereby more amount of water will be released. That is, it allows atrial natriuretic peptide releases sodium by inhibition of aldosterone, releases water by inhibition of ADH. That is the main function. Atrial natriuretic peptide, it releases sodium through the urine by the inhibition of aldosterone, and it allows the more water to go outside along with the urine by inhibition of ADH activity. So this is all about the three factors. One is the antidiuretic hormone, another one is the angiotensin, renin angiotensin aldosterone pathway. So these two pathways are get inhibited by another pathway, that is the atrial natriuretic peptide pathway. So this is all about the counter current mechanism and the regulation of kidney function. Thank you one and all.